Okay, now we're recording. Like I said, Cooper is here. Breeden is not. Elias is not here. Davis is online. Lloyd is online. Emmerich is not here. O'Brien is here. Ray is not here. Michael Baumgartner is here. Pena is not here. Sigmund is not here. Bayard is not here. Johnson is online. Huffman is not here. Hamlet is not here. Fitzpatrick, not here. Thornabar is here. Lacey is here. Tyrus is here. Byrne, not here. Hill, not here. And Sullivan. Did I forget anybody? Did I call you? Oh, okay. I was like, it's hard to say. I thought I didn't recognize you, but with you way back there and me not wearing glasses and you wearing a mask. Oh, that's right. You emailed me. All right. So before we get started, do some announcements to remind you of some things. Lab report is now past due. I'm very sad to say most of you failed it because most of you just didn't do it. Like I said on Monday, at least go in there and put your outline, title, the word abstract, the word introduction, the word materials and methods. That would have at least given you 1 point for each of those. Because remember, just like everything else in this semester, you can go back and do late work and fix it. But it's going to be an average of what your original grade was versus what you ended up with. So even if you get a 100% when you turn in your late lab report, it's going to be a 50% if you had a 0 today. Right? So why? I don't understand it. And I'm asking you, I'm begging you, if you need help, come to me. Come to me. I've got office hours. I'll meet you outside of office hours. I'll meet you in person. I'll meet you online. Please let me help you.
I have a question.
Yes.
I thought the lab report, I know it was due today, but I thought it was due at 11:59.
Yeah, 11:59. Oh, Johnson, you may be a lifesaver. Thank you. And thank you for speaking up.
Thank you. I was ready to say, I was like, I thought it was due at 11:59 tonight.
Thank you. This would be good. That would be just a good scare for me.
Yeah, me too.
You're welcome, everybody. Sorry.
Thank you, Johnson. And I'm going to give you extra credit for that too.
Yeah, me too, because I was about to turn mine in not too long ago just to read over it.
This is so great. You have no idea. You might not think it because it might seem like I'm hard, but this is really crushing me. I was like feeling so bad that I had to give everybody so many zeros. Every time I give somebody any points, I go and look at my total sheet to see how the total grades go up because I love to see it when I get fewer Fs and more Cs and Bs. So thank you, Johnson. So like I was saying, lab report is due tonight before 11:59 p.m. And if you need help, well, I hate to say this, but if you need help, visit me during office hours. But if you wait this long, it might be hard to get my help, but I will do my best. So that being said, tomorrow, after that, it is past due. And like I said, you can turn things in late. So my recommendation is if you haven't done it, like starting tomorrow, accept what you have for now and then focus on this next thing. Independent work is due Sunday. And that's the one thing you cannot go back and redo, right? So once it's done, it's done. So go ahead and focus on that. And then after Sunday, then you can start working backwards and fix your lab report, fix the labs that you've missed or that you have missed questions on, fix your absences because you know you can still fix that. All these things can be fixed. Whew. I feel so good now. Expect delays. I have one student, I feel so bad because he kept emailing me yesterday about things. I'm like, I don't know. Like I prioritize things. I have a list of priorities and I start with my oldest to the newest, right? So I'm working on things I needed to grade a while ago and emails I needed to get back to. And I warned you guys this would happen, but since independent work is due and lab reports are due, I'm getting swamped with a lot of emails because a lot of people waited until the last minute. And I'm not judging. I've been a student. Sometimes you have to wait till the last minute. Or sometimes it's because you like playing video games instead of doing work. But I don't know your situation, so I'm not judging. Any questions about this? Again, Johnson, thank you so much. I'll probably have a smile on my face for the rest of the day. You made my day. So yeah, I guess you can ignore the zeros that you have on your worksheet. They're not in the grade sheet yet. But
Okay, thank you. You're welcome.
no, thank you. All right. So let's jump into it. Oh, and then someone else asked a question, so I want to get this while we're recording. They were asking a question about lab next week. I just want to clarify because I know sometimes it's hard to decipher what's on the announcements. So for the lab this coming week, it is an online lab in that we're doing it on a computer because it's evolution. And I asked the guy who runs our lab, our core lab, who sets up those carts for us, I asked him if he had a time machine so we could visualize evolution in action. He does not have a time machine. So because of that, we're going to use computers. And like I said, we're going to do that even though it's online. We're going to be doing it here in this room. So come here like you normally would, noon on Wednesday. Bring your computers if you have one. I'll be providing some, and we'll get it done. And then we're going to stay until 1:50, which is when lab ends, or until you're done, whichever comes first. And the caveat to that is if you happen to get the lab done and I've graded it before Wednesday, then you don't need to come in. Like you don't need to come in and just sit here because if I've already graded your lab. So any questions about lab next week? All right. So for those of you online, I'm going to do things the way I've usually been doing them for the past week. We're going to use keywords. As long as you email me before I post the questions, then the keywords will work. And today, just like Wednesday, I'm going to break up those keywords. It's going to be sort of like a sentence. So the first word in that sentence is red. Anybody have any questions? Make sure you heard that. The word is red, as in the color red. All right. Good timing. All right. Perfect timing. Elias is here. Make sure you remind me that you came in at the end of class. Let's jump into where we left off. We left off with the rules of pedigree, right? The next thing we're going to talk about is family pedigrees. Your book goes into a little bit of detail, but not much. I'm not going to test you on it. Being able to do a family pedigree is not necessary for a non-biology major. However, it is good for to help get you thinking in that way, right? It does help you think in terms of the rules of probability and this idea of dominant versus recessive. And you'll see what I mean as we go through it. And when I get time, I'm going to come up with some exercises that you can do voluntarily for independent work where you do some pedigrees. I'll get back to it. Here we go. As you already know, these two Mendel's principles that we've already talked about, right? They apply to the inheritance of many human traits. As a matter of fact, it's some of these traits that we've already talked about in lab, so you should be familiar with this. And don't worry that I just skipped that last slide. It's just an introduction. So here's some traits. All right, so look at this. Freckles is a trait. No freckles is a trait. So, widow's peak is a trait. Straight hairline is a trait. So if we're talking about hairline, if we know the two different traits, this is a good reminder. See if you remember what we talked about. What would you call this? Hairline. So widow's peak is a trait. Straight hairline is a trait. But what is hair? Character. Thank you. And you do need to be able to distinguish between the two. So character would be hair, right? Is it widow's peak or is it straight? Earlobes, right? That would be the character. The trait would be free or attached. I'm not going to talk too much about this because we talked about it in lab. We spent a lot of time talking about it in lab. So we'll move forward. There's a new word that you don't know that I haven't told you in lab, and you need to know it for the exam. Wild type. I'll give you guys a time to take notes, let it sink in. It's a really simple concept. Wild type is the thing that's most common, basically. So if you remember in the lab, you had to have a hypothesis. And then we ran the experiment, i.e. we collected data. How many people had bent fingers versus straight fingers? How many people had widow's peak versus no widow's peak? How many people had a dimpled chin versus no dimpled chin, right? We did all that. And the question was, which of those traits are going to be more commonly recessive, right? Like, are we going to see more smooth chins than dimpled chins? Because remember, smooth chin was a recessive trait. And actually, that was one of them. So this is a very important distinction because, again, using the chin as an example, we'll do it again. Raise your hand if you have a dimpled chin. I know you guys are online. You can't see. There's only one person who's raising their hand. Well, the dimpled chin is the dominant gene, right? Or excuse me, the dominant allele. That is the dominant phenotypes. So Just because it's dominant doesn't mean it's more common, right? Because again, he has a dimple chin, the rest of us don't. The one that's most common is called the wild type. Could be dominant, could be recessive, doesn't matter. The fact is it's called the wild type. So know that. And that'll be a little bit more important, or rephrase that. That will be important again when we discuss evolution. Because a lot of the times when we talk about things, traits in evolution, very often through natural selection, it's the better of the two traits that you start seeing more of. Sometimes it's not. Sometimes it's random. And that's something called genetic drift. We'll talk about that later. So, any questions about this slide? Pretty simple, right? So, how did he study genetics? What did he use? Plants, animals, protists, bacteria? Plants, good. Good. So, if we wanted to study human genetics, that's a little bit trickier, right? Because what did Mendel do to study the plants? He's like, all right, I'm looking at flower color. I got a purple flower colored plant here. I've got a white flowered plant here. I'm going to make these two things mate and look at their offspring, right? You can't do that with humans. I can't be like, all right, yeah, I want to see what happens when I mix you and her, right? Who doesn't exist, for those of you online. That's hypothetical. And I can't force that. So how do we study genetics in humans? Any guesses? Yeah. I love that, because that one, even though I wrote this PowerPoint, that's not the answer, but even though I wrote the PowerPoint, that's the first thing that popped in my head, because now that is the more accurate answer. Yeah, because for those of you online, he said DNA. So you think about things like 23andMe, which is mostly ancestry, but yeah, DNA is one of the things. But another way we look at it, rather than forcing matings between this person and that person, right? All we have to do is look backwards, right? This is what geneticists do. And you don't need to write any of this down necessarily. I'm not going to ask you what a geneticist does, but just a discussion so you can think, get in the mindset of studying human genetics and how that might work. A geneticist analyzes the results of matings that have already occurred, right? Again, it's not ethical to say you mate with you and then get your offsprings and mate with their offsprings, right? We can't do that. But lucky for us, there's a lot of mating going on in the human species. And a lot of it has already happened. So we can look back and see these things that have already happened. We can't control the experiment, but we can still collect the data, right? And when we do that, we call these things a pedigree. And I'm sure you're probably familiar with it. It's basically a family tree, but instead of strictly focusing on your ancestry, like, oh, there's your great-great-grandpa who came over from Ireland or whatever, right? You're actually going to look at alleles and genes, traits, diseases, things like that. We can also use a pedigree to determine if something is sex-linked. And for now, I mean, you don't even need to write that down if you don't want right now, because we're going to discuss that later. Right now, that's just the introduction, the idea of there is something called sex-linked, and we're about to talk about it. This next figure I'm going to show you, it shows a pedigree that traces the incidence of free versus attached earlobes. I'll give you a second before we move on to it and talk about it. I might do a little bit of writing on the board, if it'll allow me. Are there any questions so far before we, while I'm waiting? No questions? That's good. No questions is good. All right, so the second word, since I'm waiting anyway, the second word for those people who need attendance online is going to be fisherman. I really don't care if it's plural or singular, because I know it might be hard to hear what I'm saying, but fisherman. Anyway, let's move forward. Let's look at this hypothetical pedigree. I know I shouldn't have to say this, but just to be sure, this is just an example. I'm not going to have a question on the study guide where I say, hey, remember that pedigree for attached earlobes? How many males were there were attached earlobes, right? It won't be like that. But let's talk about some stuff. We know, I'm going to try to do this quickly, because again, you're not going to have this on the exam. Well, maybe for extra credit. But I'm not going to make you do it. And I'm hopefully, if I have time, I'm going to make some things like this, and you can do it for independent work. But here we go. Attached earlobes. Is that a dominant trait? Yeah, dominant trait or recessive trait? You guys remember having it attached? Good. Not that you need to know that, because that was just an example, and I won't test you on that. This is not an ear class. But yeah, attached, let's just abbreviate NA for not attached. We'll just say it was dominant. Let's just do big E, right? For dominant. A for attached, let's call that little e for recessive. You guys with me so far? Okay. So we know, because they told us, anything that's purple means they have the recessive trait, right? They have the attached earlobe. Meaning their genotype has to be what? You can either give me the name for it or give me the letters. Here's your choices. And I'm going to abbreviate here again. It's a multiple choice. 3 choices. Homozygous dominant, heterozygous, or homozygous recessive. Somebody who has the attached earlobes. Which of these 3 are they?
Recessive.
Good. Homozygous recessive. It's a good time to remind you that if you show a recessive trait, then you have to be homozygous recessive, because both of your alleles have to have recessive. Because if you have one of those dominant alleles, it's called dominant for a reason, right? It's going to dominate. And you're going to have that trait instead. So there we go. We know anybody who has an attached earlobe is homozygous recessive. So let's go ahead. I'm going to write that down on there. So we know this person is little e, little e. We know this person is little e, little e, little e, little e. Couple right here. All right. Now, let's take a look at, I don't know, let's start here. It's probably easier and it's at the top. So that's why I chose to start here. We know these 2 people's genotypes, but we don't know these people's genotypes. We do know, because they have the dominant allele, we know at least half of their genotypes, right? We know they're at least one big E. They're big E something. Big E something. We just don't know what the something is. It's almost like when we talk about those Labradors, and where we mated this one with the recessive traits, and that's how we would have been able to figure out that unknown genotype. So let's try to figure out this person. Are they big E, or are they big E, little e? Before I show the work, is anybody just good enough where they can see it without actually having to write it out?
Heterozygous.
Yes, heterozygous.
Because they can't have a kid that's recessive. They have both the dominant.
Exactly. The reason we know that this person has to be heterozygous, and I'm focusing right here on this recessive allele, is that this person, or this couple here, has a kid who's homozygous recessive. Meaning they have 2 recessive alleles, meaning they got 1 recessive allele from the dad and 1 recessive allele from the mom, right? So we know that just has to be the way, right? Moving forward, now with that being said, we know these people's genotypes too. We know neither one of them are homozygous recessive, otherwise they would have the trait. So we know they're both big E something, right? And we know again, because they have 2 kids that have 2 little e alleles, 2 recessive alleles, that both of them have to have at least 1 recessive allele. See? And that's how, I'm not going to go through all this. And that's how a pedigree works. Of course, I'm so nerdy, I find that kind of stuff fun. So when I was in genetics, I actually enjoyed doing that stuff. Until it came time to turn it in for a grade. It's a little less fun when you get graded on it. But, in your situation, like I said, it'll be independent work, so you won't have to do that. So if you do it, there's no pressure, because if you get a bad grade, it's just extra points for independent work. Thank you. Okay. So any questions about pedigrees? All right. And again, I might have 1 on there on the exam, but only for extra credit. All right. Let's move forward. Let's talk about human disorders caused by a single gene. And before we jump into it, I want to remind you, for the most part, everything we have talked about is caused by a single gene, or at least that's the way I've taught it. And remember, I told you in the lab, I gave you a little caveat that some of this stuff isn't as simple as we've made it out to be. But for the sake of the lab and getting hands-on experience with this stuff, we simplified it. But yeah, let's talk about this. Human disorders caused by a single gene. Just like everything else, if we're talking about things that are caused by a single gene, some are dominant, some are recessive. In this case, again, we're talking about things caused by a single gene, right? There's one gene that causes this or that or the other thing. We're also talking about disorders that are located on autosomes. Not that you definitely don't need to write any of this down. This is just an introduction. I'm just telling you what we're about to talk about. And to jump farther ahead, because I'm giving you a preview, if you will, later, once we're done talking about this, we're going to talk about more than single genes, right? And then we're going to talk about things that are located on sex chromosomes instead of just autosomes. Here's a table from your book. You definitely don't need to memorize it. Just some examples here. Here's some recessive disorders listed here. Some dominant disorders listed here. This tells you the name of the disorder on this side. That tells you the major symptoms. You guys can look this up if you want for independent work. And then this could be a very short paper if you want it to be like, okay, what is sickle cell disease? You can look it up and probably get a very quick answer where it says that, it's a recessive disorder and it causes, it's caused by one mutation in the blood cells, and you have, because of that, it's misshaped. Go on, and you can just leave it at that, or you can get into more detail. That's the beauty of this for independent work. Make it as easy as you want. But anyway, this is just a table. There's nothing to memorize, but we are going to talk about some of these, and let's do that. Here we go. First thing we're going to talk about is recessive disorders. So again, we're still talking about things caused by a single gene, and now we're looking at recessive disorders. And before we talk about it, in your mind, you should just think this is the same stuff we talked about when we talked about recessive traits. So attached earlobes, smooth chin, no freckles, right? That's a recessive trait. These are recessive traits too, except now we're getting a little bit more specific. Yes, they are recessive traits, but specifically they are disorders. So all the things that you learn about how those recessive traits work, it's the same thing with this, except again, these are disorders. So they're not just like, uh-oh, your earlobe's attached, right? Some of these are bad. Most disorders are in fact recessive. Specifically, as your book says, most human disorders are recessive. Another good independent work topic if you want to look that up. What about other animals? What about other organisms, plants, protists, fungi, bacteria? Anyway, individuals that have a recessive allele but appear normal are carriers of the disorder. Now, prior to this discussion, this bullet point right here, you would have just called that, based on the abbreviation I had up there earlier, a heterozygote, right? You just call that person a heterozygote because they're carriers, meaning they have at least one allele, but they don't have it. And again, this is recessive, just like the thing for the smooth chin. So for example, if your genotype, if you guys remember, was big D, little d, you had the dimple, right? Yes, you had that one allele for the recessive trait, but because you had that one dominant allele, you didn't show the recessive trait. So we don't use this terminology, but if you were heterozygote in terms of your chin, you're a carrier, right? So I'm a carrier. The only reason I know this is because my son has a dimple chin. So, and my wife also has a smooth chin. So I have a smooth chin, my wife has a smooth chin, my son has a dimple chin. Therefore, she and I both must be carriers, right? Because we both gave him a recessive. Wait, I got that backwards. I'm sorry. Because it's a dominant trait. So any questions about what we're saying so far? Again, everything I'm about to tell you is everything you've already learned about recessive phenotypes, recessive traits, recessive alleles. It's the same thing, except now we're just calling it a disorder. And because of that, we don't just call you heterozygote. Now you're a carrier, right? You're a little bit more dangerous than just somebody who may or may not cause somebody to have blue eyes or a dimple chin or something like that, or a non-dimple chin. Here's an example of a pairing of a heterozygote and a heterozygote. And you've already done this and you should be familiar with it. You should also be familiar, because this is a monohybrid cross and we're talking about two heterozygotes, you should be familiar with that 3-to-1 ratio, right? Meaning you have, if these hypothetical people were to have kids, you would expect 3 of them to be hearing and 1 of them to be deaf, right? That's the phenotypic ratio. Everything's just the same as it was in the lab, right? And then we look at the genotypic ratio. It's also the same as you learned in lab. There's 1 homozygous dominant. There's, there we go. So we'll call that 1. We have 2 heterozygotes right there, right? And we have 1 homozygous recessive. Oh, that looks horrible. You guys get the picture, right? It won't let me do it. Anyway, we're still looking at a 1 to 2 to 1 ratio, right? Because this is just the same as everything you've already learned. Now we're just adding some words to it. So again, if you are heterozygote in the case of a recessive disorder, we call you a carrier. Pretty simple, right? You're a carrier because you don't show the disorder, but you have the allele for it. Any questions about that? Okay, this is a good time to remind you of something I told you earlier in the week. These are just probabilities. So in theory, this couple here, this heterozygote and this heterozygote, could have 10 kids. It would be very improbable, but they all could be homozygous dominant, or they all could be deaf. Very unlikely, but it's possible. These are just probabilities. So if they have 4 kids, even though you might expect 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive, those are just probabilities. It does not always work that way. So any questions about that? All right, let's give you an actual example from your book, cystic fibrosis. The reason that is the example your book uses is because it is the most common lethal genetic disease in the United States, at least at the time that this book was published. There's another really quick independent work idea for you. Confirm that for me. I don't know. Is that still the most common lethal genetic disease in the US? I assume it is, but I don't know. It is caused by a recessive allele carried by about 1 in 31 Americans. This is just an example. You do not need to know any of that. It's just an example. Your book even goes on to tell you what cystic fibrosis does and all that. But this is a long chapter, and I'm not going to spend time on that. This is not physiology and anatomy class. I'm not teaching you about diseases. Specifically in this chapter, I'm teaching you about genetics. So you can look that up if you want. But as far as I'm concerned, that's just an example. I'm not even asking you I'm not even asking you to give me an example of a recessive disorder. But there you go. There's cystic fibrosis. Any questions? All right, nice and easy. That means we're going to talk about dominant disorders next. We're done talking about recessive disorders. Let's talk about dominant. I'll remind you, when we talked about recessive disorders, I did say first, right off the bat, that recessive disorders are more common than dominant disorders, right? Can anybody guess why? Actually, we'll come to that. Let that marinate in your head for a little bit, because I'm going to come back to that. But start thinking about that. Why would recessive disorders be more common than dominant? But let's talk about dominant disorders. Your book already went through the whole spiel of being a carrier versus a non-carrier. Obviously, that's not the case with dominant disorders, because if you have that one allele, then you have the disorder, right? So you can't be a carrier. If you have one allele, at least one, you've got the disorder. But you already know the rules of probability. You already know Punnett squares, right? So just keep that all in the back of your mind as we move forward. The first example your book gives is achondroplasia. It is a form of dwarfism. Again, it's just an example, just like cystic fibrosis. I won't ask you any questions about this on the exam. Nothing that you need to memorize. There's a caveat to this though. So if any of you have looked at the study guide yet, there is a question about this on the study guide, but it gives you all the pertinent information. So it tells you everything that you know here that I'm about to tell you. And then it asks you to say, all right, if this person mates with this person, what's the likelihood of their kid having this or not having this? So keep that in the back of your mind too. But here we go. Achondroplasia. Again, it's a form of dwarfism. I like that they chose this one because to me, it's a more interesting situation. Because again, as you know from dominant traits, right? Even if we're just talking about widow's peak or freckles or chin dimples, you know that all you need is one allele and you have the trait, right? You can be homozygous dominant, heterozygous, doesn't matter. If you have the one allele, you have the trait. Achondroplasia is slightly different in a way, and I like it. Well, I like that we're using this as an example. So first of all, the homozygous dominant genotype, right? Big A, rather than just having it, it actually causes death, right? So if you had, I don't know, a heterozygous person that mated with another heterozygous person and you would do the Punnett square, right? You would see there's a 1 in 4 chance that, your child would be homozygous dominant, i.e. there's a 1 in 4 chance that your child would not even make it to childbirth. And again, this is just an example. I'm not going to ask you these details. Because of that, it's only the heterozygotes that have achondroplasia. So this is an interesting situation. Instead of just having one allele, you can only be heterozygotes because, again, having both alleles, both dominant alleles, being homozygous dominant, it's deadly. So if you have achondroplasia, like this person here is one of the more famous people who has it, then your genotype must be heterozygous. Because if you were homozygous, you'd be dead. Excuse me. If you were homozygous recessive, you would never have been born. And obviously, if you were homozygous, it's good grief. I'm gonna drink more coffee, slow it down. If you are homozygous dominant, you would never have been born. If you were homozygous recessive, you just wouldn't have the disorder. Therefore, to have the disorder in this situation, you have to be heterozygous. Meaning, and this is just an example too, again, nothing to memorize here, but a person that has achondroplasia, who again is a heterozygote, has a 50% chance of passing that condition on to their children. And I'll show you why. Let me show you the work behind that, the math, so to speak. Here we have somebody without achondroplasia mating with somebody who has achondroplasia. We know that the person without achondroplasia, without even looking at their genes, we know that their genotype has to be homozygous recessive. Otherwise, they would show the condition. We know that this person who has achondroplasia must be heterozygous because if they were homozygous dominant, they would be dead. If they were homozygous recessive, they wouldn't have it. So anyway, this person without achondroplasia, they go through meiosis, right? Those different alleles split up, right? So now 50% of their sperm have little d, 50% of their sperm have little d. Another way of saying that is 100% of their sperm have little d. This dwarf, this person with achondroplasia, they go through meiosis. Their alleles split up. 50% of their eggs have big D. The other 50% have little d. Again, by the rules of probability, which we learned last week, we know that we can have, there's a 1/4 chance that we'll have this situation, right? Big D, little d. Another 1/4 chance that we'll have this. And of course, 1/4 plus 1/4 is 1/2. The only reason I'm going through this so quickly is because we've already talked about this. That's the only reason I'm just like giving you the math. You already know this. So it's like 50% chance that this will happen, right? And then obviously 50% chance that this will happen. But again, this is just an example. There's nothing to memorize here, but you understand how this works, right? And heads up, there will, again, probably be a question on the exam where I give you all this pertinent information, right? Again, something like this. So you don't need to memorize, all right, homozygous dominant is deadly, heterozygous has the condition, and homozygous recessive doesn't. You don't have to memorize that. I'll give you all this information, and then there'll be some situation where I say, okay, what are the chances of 2 dwarfs mating and having this? Or what are the chances of 1 dwarf mating with a normal person and having that? Or I could get really complicated, and let's go ahead and do it right now, because when I say really complicated, I'm being slightly facetious, because it's only a step above this complication. If you have a dwarf mating with a non-dwarf, as we have in this situation, what are the chances that they will have a boy who has achondroplasia? Right? So we already know that there's a 1 in 2 chance that they'll have achondroplasia, right? But what are the chances that they have a boy instead of a girl? You guys don't overthink it. Forget all this. Purge that from your brain for a second. 50%. Thank you. 50%. 1 in 2. So 1 in 2 chance. 1 in 2%. 1 in 2 that they'll have a dwarf. 1 in 2 that they'll have a boy. By the rules of probability, then, if you were to get that question, you would know that you would need to multiply these for this thing to happen and then this thing to happen. There's a 1 in 4 chance, right? Because you multiply it. 1 times 1 is 1. 2 times 2 is 4. Pretty good. All right. Any questions about this before we move forward? So again, I like achondroplasia as an example because it's one of those interesting situations where the homozygous dominant is actually deadly. It doesn't exist other than for a little bit in the world. But the next one we're going to talk about is the most dominant lethal disorder, or one of the most dominant ones. It is Huntington's disease. And again, we come back to this idea here that dominant lethal disorders are much less common than lethal recessive disorders. Why might that be? Why is it that we see many more lethal recessive disorders than lethal dominant disorders? Keyword there is lethal, I think.
Because they get passed on.
Yeah. If you have a lethal dominant disorder, it's gonna kill you, right? Because it's dominant, there's no carrier. You're gonna be homozygous dominant, you can be heterozygous. Either way, you have it, you're gonna die, and most likely you're not going to pass, or you have less chances of passing it along, right? Because it's lethal. And you have more of a chance of having it because you could have two genotypes. Either genotype will give it to you. Unlike a homozygous or recessive disorder, where you have to be homozygous recessive to get it and for it to kill you. So it's easy. You could be a carrier and be non-affected and pass it along. So yeah, there you go. Anyway, Huntington's disease. Again, your book gives you some information about it. That's a degeneration of the nervous system. It usually doesn't begin until middle age. Once that happens, it's usually irreversible and fatal. Because the allele for Huntington's disease is dominant, i.e. because it's a dominant disorder, any child born to a parent with the allele has a 50% chance of inheriting the allele and the disorder. Again, just like cystic fibrosis, this is just an example. I'm not going to ask you any questions about this on the exam. But this is something to think about. This is slightly interesting. So in my opinion, these two bullet points kind of go together right here. So yes, dominant ones are more, less dominant lethal disorders are not as common as the recessive lethal disorders for the reasons we already talked about. So Huntington's disease, it's interesting that this is one of the more common ones because it makes sense. Because you don't even start getting it until middle age, there's a higher probability that you did survive to have kids and pass along that allele, right? You haven't died yet. So that is probably one of the reasons that Huntington's disease is relatively prevalent. Any questions about that? All right. Again, I teach out of the book. Every chapter has this thing called the process of science. I'm always going to skip it. There are questions about it on the study guide. I won't ask you any questions about it on the exam. I highly recommend reading it anyway because, again, it's a good brain exercise to get your mind thinking scientifically. Plus, if you like dogs, or dags, as they said in, what movie was that? Snatch. Look that up if you want. Anyway, if you like dogs, you might be interested in this. It just talks about all the different varieties of dog hair. I'm going to go through it. Again, not going to test you on it. But if you happen to read it and have questions about it, want to talk about it, let me know. So here we go. We're done talking about recessive disorders. We're done talking about dominant disorders. Now let's talk about something called genetic testing. To me, this one's really interesting. As technology advances, first of all, genetic testing becomes a little bit easier, becomes a little bit cheaper, and there's more implications. And your book doesn't get into this too much, but I'll talk a little bit more about it when we get there. But let's talk about genetic testing. What is genetic testing? It's tests that detect disease-causing alleles. So the stuff we just got finished talking about, those disorders, the dominant and recessive disorders, well, there's ways to test for it. Most of these tests are performed during the pregnancy if the parents are aware of an increased risk of genetic disease, right? So if you know that this runs in your family or that runs in her family, yeah, your doctor might say, we should probably get you a genetic test. Or going back to something we learned previously, remember that whole trisomy 21 thing and advanced maternal age? A lot of times if a woman is over a certain age, they will do this. In a sense, that is genetic testing. It's a little bit easier though, because then you're just looking for an entire extra chromosome as opposed to specific genes. Anyway, let's talk about some of these tests. I'm not going to test you on this. I'm not going to test you on the test, so to speak. Amniocentesis is one of them. I'm sure you've heard of it, or maybe you've heard of it. That's when they use a needle, stick it in, pull some fluid out, and look at the cells in the fluid. Your book gets pretty specific. 2 teaspoons. I thought it was interesting that they'd be that specific, but then also use teaspoons as opposed to milliliters, but whatever. So that's all the book, and that's all the information the book gives you, or most of it. I'm not going to test you on that. Look it up if you want to for independent work, if you want to look deeper into it. Here's a picture of it in action. I don't know if you can see, but this hand right here is holding the needle. What's this hand right here doing? What is this thing? I'll give you a hint. What?
Is it a syringe?
No,
That's a tube.
it looks like a tube. Any other guesses what this is? And there's your hint.
That's a sonogram.
Yeah, sonogram, or what do we call it now? Ultrasound, right? And why would you want that? Why is he doing that?
To see what he's doing.
Yeah, to see what he's doing, right? You don't want to just go poking in there and hit the kid. Probably not a good thing. So yeah, anyway, there's a picture of it in action. Here's a drawing of it in action. I think this is from your textbook. I don't remember. Anyway, there he's got the ultrasound and pulling out some fluids. You could give that a completely different caption and make it horrible. Like, there's a party, playing some music for the kid, giving him some drugs. Just kidding. All right, any questions about this? All right, the next example that your book gives, and I'm not going to require you to know anything about, is CVS, chorionic villus sampling. It's similar. You're still trying to get fluids out. You're still looking at the cells in the fluid. It's a newer thing. Instead of using an actual needle, it's a more flexible tube that is inserted into the uterus. But this time it goes for placental tissue. Again, I'm not going to ask you about any of this. The point is, we know that these genetic disorders are out there. We know that some people are at higher risk than others. So what do? We do tests. Yeah, blah, blah, blah. Talk about that. So here's the real question then. What do you do then? What happens with the test? How's that useful? What happens if you do the test and you find out, yep, sure enough, this is going to have Huntington's disease? What do you do?
To help you make, in my mind, it'll help you make decisions on if you're going to go through the pregnancy or abort.
Right? It's a very personal answer that I won't even get into. But yeah, like for some people, it's like, it might be a question of, do you abort this or do you have it? Or for other people, it's a question of, okay, we know that this kid's going to have this. What can we do in our lives to prepare for that? So that's why one of the reasons genetic testing is important. And I also think it's important to note, I don't think your book does, is these days with IVR, which is a form of, like if you can't get pregnant, you can do it artificially. And it's slightly different than, I don't want to use artificial insemination, but basically you take a bunch of eggs and a bunch of sperm and you do it outside of the body, you fertilize them. And then you're hoping that at least one of them works, and then you can implant that into the female. But sometimes, and this was the case with my son, sometimes more than one takes, right? And then you got to decide, well, and this is still when they're just a zygote, right? Still a few cells splitting. What do? Do you keep them all? Do you keep one of them, right? So just something to think about. And I think if I haven't shared it yet, I will share it with you. This is where this idea of designer babies comes in, because what you could do, and I don't mean you could do this ethically, but I mean scientifically this could be done. You could always do IVF, right? You take your sperm or the dad's sperm, the mom's eggs, you do everything on a petri dish, so to speak, and then you examine each one of the eggs or each one of the zygotes and decide which one you want based on genetics. And some people might do it based on something other than disorders. Like, all right, well, this person's more likely to have this or that. And the more we learn about genetics, the more likely that is. Like intelligence, we don't know the genes responsible for that. We do know it's genes and environment. But what happens once we nail down all the genes? Then what? Are people going to do that? And they're going to look at these eggs and say, all right, well, of all these zygotes, this is the one that's genetically more likely to be intelligent than the rest. Or this one's the one that's going to be genetically more likely to be an athlete. Or this one's more genetically likely to be a musician. So that's something to think about. There are real implications, especially for you younger people. You know, this might be in your lifetime or your children's lifetime where that might be something offered to the richer people, or who even knows, maybe even poorer people.
It sounds like a Build-A-Bear type of thing.
Yeah, it does.
Because when you said design a baby, I'm like, it sounds like a off-brand Build-A-Bear. But it also sounds like one of those like flash games you get like an ad like design your own baby.
Yes, yeah. And it is, it's similar to that. And again, too, this is just the scenario I just described is you or somebody choosing which zygote makes it, which doesn't. I didn't even talk about this idea of gene editing, which we'll talk about later. Then it's less choosing which ones have made it naturally. Then it's more like, all right, I'm going to delete this gene and add that gene and delete this allele and add that allele. Like, it's crazy to think about what is possible. and what probably will happen within our lifetime. Anyway, let's move forward. So it's Friday, right? We've been doing this since Monday, and we have just now finished the first of the 3 main bullet points. Such a long chapter. So let's jump into it. We've only got 6 minutes left, but normally I'd be like, it's Friday, let's head out. But we need to jump into it because, again, we should be done with this chapter by now. So remember, everything we just talked about, whether it's the traits, chin, eyes, freckles, one gene, right? All this is these disorders we talked about, one gene. But here's a little thing that you should keep in mind. That's not usually the case. Usually, it's more than one gene involved. So usually there are variations on Mendel's laws, and that's what we're about to talk about next. So again, everything we just talked about prior to this is not the normal scenario necessarily. It's not the more likely scenario, but it is the more easy to teach because it's a lot less complicated. So you need to understand that before you understand this. The next word in the sentence, for those of you who need attendance, is going to be pickup truck. Anyway, let's talk about these variations. Mendel's laws explained inheritance in terms of genes passed from generation to generation according to the rules of probability. You already know that. We said that in the first main bullet point. I'm saying it again. So this is still the case. The rules of probability still apply, at least for all sexually reproducing organisms, right? It's a little bit different for things that reproduce asexually. However, Mendel's laws do not explain certain patterns of inheritance. So that pedigree I just showed you, or not just showed you, there's one I showed you earlier about the earlobes, that can be applied to anything we've learned so far. Anything that's controlled by a single gene, dominant or recessive, we can figure it out. It's very simple. But again, most things aren't controlled by a single gene. Therefore, they don't follow those same patterns that you would see on a pedigree that we saw earlier. So any questions about this slide, which is just the just an introduction, just a little preview about what we're about to get into. All right, here's the first thing we're going to talk about. Incomplete dominance. We're only going to talk about it in plants and people. Obviously, there's other organisms out there we don't talk about, so you can look it up if you want. Protists, fungi, so on and so forth. But before we talk about it, I want to let you know we are still, at this point, still talking about a single gene. So yes, this is a variation of what we talked about earlier. Things are getting a little bit more complicated, a little bit less simple than they were earlier, but we're still talking about a single gene. So we know, you guys already know this idea of dominant and recessive, right? If you have a dominant allele, it masks over the recessive allele, right? So if you have an allele for the dimpled chin, doesn't matter what the other allele is, you have a dimpled chin, right? Now here's a word I haven't told you before I get into this. From now on, now that we're at this point in the chapter, all that stuff I talked to you about earlier, I'm going to call that complete dominance, right? So yes, I've been just calling it dominant and recessive, but every situation I told you thus far is something we call complete dominance. Now we're going to talk about incomplete dominance. And basically, and I know that's all written on the board there, but look, incomplete dominance, instead of one allele beating out another, it's just simply that they mix. That's all you need to know about incomplete dominance. It's not one beats the other, it's that they mix. So in the dimple chin, that would be hard to tell. But technically in that situation, if you were heterozygous, you, instead of having a dimple chin, you would have a slightly dimpled chin. Obviously that doesn't work, right? Same with freckles. But this is a great example. If this was a complete dominance situation, you can see that that's a big R, right? So in a complete dominant situation, all these offspring would have been red, right? Because they would have all been heterozygous. But in this situation, the heterozygous, it's a mixture of both phenotypes. That makes sense? So red is not completely dominant to white. It is incomplete. So you mix the two. And that's a great place to stop because that is simple. So if you let that just soak in for the weekend, then we come back on Monday, I give you something similar but different. And that'll make things more complicated. So again, for those of you online, yes, I've already called you out And I know that you were here online, but remember, you need to email me and give me those words that I told you throughout the lecture. Any questions? Johnson, thank you again. You're a hero to your classmates, and you really made my day too.
You're welcome. Thank you.
Okay. Basically, you got your genotypes, and it's basically one for one. One